So good morning, everyone, and thank you for coming to this morning session after the wild party in after hours yesterday. I'm Ornit Shibafalek from Duke University at the US, and with me, Byron Chris from Brunel University at the UK, co-chairing this session on behalf of the Multiomics work group of PIA. Today's session will feature multi-omic approaches to understanding clinical and neuropathological heterogeneity in Alzheimer's disease.
Before we get started with the speakers, I just want to remind everyone that Q&As can be submitted via the online conference platform or the in-person app. Questions are going to be answered at the end of the session, so please include who your question is for. Questions are anonymous as well, and so make sure you include your name if you would rather not be anonymous. So with that.
All right, so we're going to start with our first speaker, Pablo Garcia Gonzalez from ACE Alzheimer's Center in Barcelona, the International University of Catalonia in Spain, and he will tell us about integrative multi-omics analysis reveals physiological and genetic driver of CSF biomarker variability implication in neurodegeneration studies.
Okay, thank you for the introduction, and also thank you to the AIC for supporting my attendance to this conference. So CSF biomarkers are central to neurodegeneration, and omics studies have become quite prominent to disentangle disease complexity and identify novel biomarkers. CSF omics data are characterized by high redundancy, forming large correlation clusters, and studies similar to ours have identified large amounts of inter-individual variability in their overall protein levels. However, the underlying factors driving this observation still remain poorly understood. So in this study, we analyzed a memory clinic cohort of almost 1,400 individuals based on mainly MCI or dementia subjects, but also a small subset of cognitively unimpaired individuals. And we analyzed 3 data sets: a lipidomics experiment measured by mass spec, a proteomics experiment measured with ExomaScan 7K panel, and a genomics data set with TopMed imputed genotypes available. So the image you can see here represents the standardized values of every lipid and every protein species that we measured. And as you see, there is a pattern that is quite evident in the data, where some samples display higher or lower overall lipid and protein levels than others. And when we actually look at the relationship between the intensity signal in the lipidomics and the proteomics experiment, we observe a really strong correlation, which indicates us that this pattern is caused by an intrinsic property of the CSF and not by experimental noise. So we performed a principal component analysis to disentangle this variability, and we found that just with the first 2 principal components, we can explain 75% of the variance in the proteome and the lipidome in an experiment. And as you can see, also PC1 and PC2 were quite consistent between both omics datasets as well. Also, we ran lasso regressions, and we found that these 2 PCs were also the best predictors of the global intensity signal we were quantifying in the previous step. So we then performed a more thorough characterization by running multivariate linear regressions, and we found out that the PC1 was really strongly associated to CSF phi-tau and A-beta42 levels measured independently with ELISA or Lumipulse. And importantly, this effect goes in the same direction, indicating us that it follows the concentration axis. So another way that we can interpret these results is by understanding that phi-tau and A-beta42 are among the proteins that are affected by this inter-individual variability in overall protein levels. And on the other hand, for PC2, what we found was a really strong association with CSF turbidimetry, which is an indicator of the total protein amount in the CSF, and is used often as a proxy of blood-brain barrier damage. So of course, because we were analyzing a memory clinic cohort, we wondered whether these PCs were capturing disease heterogeneity, but with the exception of the proteomics PC2, which was increased along the dementia continuum, we found that the PCs were independent of disease status and disease progression. So then we ran a GWAS analysis of these cryptic phenotypes, and we found that the same locus was associated with genome-wide significance with the intensity and the PC1 signal in both the proteomics and the lipidomics datasets. This gene, GMNC, is a master regulator of the differentiation of multiciliated cells and is highly expressed at the choroid plexus. So this already starts providing an explanation of what may be going on here. And if you look at this diagram, you see that both the epithelial cells of the choroid plexus, which are the cells driving CSF secretion, and the ependymal cells, which are also involved in CSF production and distribution across the CNS, they all express abundant cilia in their apical surfaces. So in some way, this is connecting CSF production, CSF distribution, and likely CSF clearance with the global intensity signal that we are observing. This locus is also the most strong locus associated with lateral ventricular volume, and what we observe is that it was a general pattern with the alleles increasing ventricular volume consistently associated with a decrease in these cryptic phenotypes. And also the CSF levels of p-tau, A-beta40 or A-beta42 were also consistently decreased by these alleles. Not only this, but when we tested the association of these alleles with the individual protein and lipid species, we found really a strong inflation in these models with very asymmetric volcanoes and a tendency for the alleles increasing ventricular volume toward downregulation of the protein and the lipid species. And finally, we validated our results in an independent cohort with available proteomics and metabolomics in CSF, plasma, and brain, and observed a very similar pattern that was exclusive of the CSF omics data. So basically, this led us to general observations where you can see that genetic predisposition to larger ventricles leads to generally lower CSF concentrations. But as I'm about to show you, the pattern is a little bit more complex, and we actually could detect three distinct proteomic signatures, one related to PC1 upregulation, another one related to PC1 downregulation, and another one related with PC2 upregulation. And we performed gene cell enrichment analysis and gene expression profiling to help us interpret these signatures, and also integrated this with the associations found with the ventricular volume increasing alleles. So based on this integrative analysis, we reached to the conclusion that PC1 is likely driven by CSF turnover rates, and here you can see how consistent the association of the ventricular volume increasing alleles are in the top 25 upregulated and downregulated proteins, and they always go in the opposite direction as the PC association. So PC1 upregulated proteins, which comprise more than 62% of all the proteins measured in the SomaScan dataset, are likely proteins generated in the brain and more likely diluted in individuals with higher CSF clearance. And on the other hand, we see a small subset of proteins that are actually more concentrated in individuals with higher CSF clearance. And these included proteins such as leptin or prolactin, which are known to be transported from the blood to the CSF by the choroid plexus. So it makes sense that they have increased concentrations with higher CSF production. On the other hand, the PC2 signature was not associated with ventricular volume increasing alleles, indicating a different biological background. And what we found is that the top upregulated proteins by these PCs were mainly plasma-abundant proteins synthesized by the liver and included coagulation factors, complement proteins, and apolipoproteins. So we concluded that this PC was likely caused by brain-blood barrier dysfunction. And also an interesting finding was that the proteomics PC2 is very strongly associated to age, male sex, dementia continuum, as we showed earlier, and also to a patient history of BBB disrupting disorders, resembling the connection between these disorders, BBB damage, and dementia. So importantly, our findings have two major consequences. One is that absolute biomarker levels in the CSF are affected by these cryptic phenotypes, and this has important consequences, for example, when we try to perform cutoff-based classifications, such as in the ATN framework. And also, these findings explain the strong patterns of intercorrelation of the CSF, which actually lead to strong inflations when, for example, we try to find out the proteomic signature of CSF p-tau or beta-42. And as you can see here, only by including the PC1 as a covariate, we already can reduce this inflation by quite a lot. However, we wondered if there are any more optimal reference proteins than just adjusting by the PC1. So we again scanned the SomaScan dataset to find for optimal reference biomarkers. So basically, we ranked each somamer based on their ability to increase the predictive performance of CSF p-tau and beta-42 in 2-year MCI to AD conversion, and also in their ability to reduce the lambda inflation in the proteomic signatures of p-tau and A-beta. And here you can see that the top 5 reference proteins, they all outperform PC1 as an adjustment variable. And you also can see how they correlate strongly with PC1, indicating they are proteins that are reference proteins for CSF clearance, but they don't really correlate with PC2. So this slide is to show you the profound impact this reference protein of CSF clearance has in AD biomarkers in the CSF. And it's important to highlight that this reference protein is independent of age, sex, ApoE status, baseline mini-mental, disease status and disease progression, or pretty much any other disease phenotype we could think about. And you can see how the median levels of A-beta-42 and p-tau are so strongly affected by this marker of general CSF concentration and CSF clearance. And these findings also help us explain some of the intermediate phenotypes found with CSF biomarkers. For example, A-negative tau-positive subjects tend to be in the high CSF concentration group, while A-positive tau-negative subjects tend to be in the low CSF concentration group. And finally, I'm just showing this slide to show you the potential for adjustment. Here you see the relationship or the integration of the proteomic signature of p-tau and A-beta42 if when we still do not adjust by this reference protein, and just by including our reference as a covariate, we see a much more cleaner and interpretable signatures, revealing the proteomic signature of mutation carriers in autosomal dominant AD, and also revealing some of the proteins that are known to be associated with the APOE genotype. So finally, I just want to state my conclusions and important messages. We found that CSF turnover rates and BBB integrity explain 75% of the omics variance in our data set. These patterns were really consistent across 5 independent experiments, and most importantly, the message to the audience is to highlight the importance of adjusting for these latent factors whenever we perform CSF omics studies, either by using reference proteins, which might be the optimal approach, but they still need validation, or by adjusting by latent variables such as principal components or surrogate variables. So thank you very much for your attention, and I'll be happy to take questions in the Q&A.
Thank you, Pablo, for the great presentation. And our next speaker is Alvin Thomas from Washington University in St. Louis, US. And the title of his talk is Plasma Proteomics of Cognitive Resilience of ADRD, the ARIC study. Thank you.
Hello. I would like to thank the committee for the opportunity to give this talk. I will be talking today about cognitive resilience in this session on clinical and other measures of heterogeneity. I just joined NYU about 6-7 weeks ago, so that's why now it says NYU. Let's see. Okay. I have nothing to disclose, but I would like to acknowledge the role of the NIH in supporting this work. And I'm going to dedicate a few slides at the beginning to talk about cognitive resilience, because I hope that a lot of the people in this room are doing multi-omics kind of research, given that most of the cognitive resilience talks were on Sunday. So I want to drag you into this field to get help analyzing cognitive resilience with genetic and other layers of omic data. So this is a graph we see all the time, a version of the Jack graph where we're looking at increasing levels of pathology over time. And in the original figure, this was the general layout where that last line is cognition. They updated it a few years after that first publication, and that update focused on this heterogeneity in cognition for a given level of pathology. And what this update really emphasizes is that some people with the same measurement of biofluid biomarkers could have very different levels of cognition, some being cognitively normal, some being mildly impaired, some being very impaired. After several decades of looking at cognitive resilience, we still don't know a lot about the biology, but we have continued to identify many different mechanisms that could support the underlying theory of cognitive resilience and related phenotypes like cognitive resistance, brain reserve, and brain maintenance. Tomorrow's plenary will have a section on brain resilience that I'm sure will be very exciting to attend. So there are several barriers when it comes to understanding cognitive resilience. A lot of them that we talked about on Sunday were related to measurement. It's a latent variable. We don't have a direct way to measure it, and we're left with a variety of methods that all have strengths and weaknesses. When we're looking on the omics side, this has significant impact because it lowers our ability for discovery. Often these data are multimodal, requiring more than just the measurement of a biomarker or measurement of cognition. As you'll see in this study, we incorporate imaging and cognitive data in order to measure cognitive resilience. And then, of course, we have to have the omic or genomic data as well. And sample diversity also plays into the impact when it comes to studying cognitive resilience. So what we tried to do in this study was improve on some of the diversity by including a large sample of African Americans from the atherosclerosis risk and community study, which is a community-based, originally cardiovascular study that recruited in 4 communities around the United States. At visit 5 of that cohort study, there were a subsample that went through MRI, and we also have SomaScan 5K in all those individuals, a battery of cognitive psychometric tests, and we use that data in order to develop a measure of cognitive resilience and then identify proteins associated with cognitive resilience. One of the things I want to emphasize in this analysis that perhaps we can talk about in the Q&A is that we, given the birth of clinical data, we investigated some systematic factors, including kidney function, instead of just trying to adjust for it using principal components. We did compare the 2 approaches, and we do think there's a lot of value of actually using kidney function when you're studying plasma biomarkers in this setting. So as I mentioned, ERIC is a community-based cohort. It's ongoing, and at visit 5, we had 10 psychometric tests. We used 3T MRI, and we have a well-documented set of covariates. And we defined cognitive resilience using what's called the residual approach. Essentially, we're using linear regression, and we're using the residuals from that regression to identify individuals that are either doing better than expected given their level of pathology or worse than expected given their pathology. It's not perfect. There are definitely a lot of issues with this approach, but we did do a pretty deep dive into the validation of this measure, making sure it was correlated with things defined by theory, before proceeding on to the omic analyses. Cognition used a general factor score. Dr. Alden Gross helped develop this. He's here if you want to ask him more about it. And for the MRI features, we used an a priori approach based on prior work that identified specific elements of the MRI data to incorporate. We did also use machine learning, both from random forests as well as some neural networks. There was a lot of concordance between these methods, and we ultimately proceeded with what we defined a priori. Importantly, and in contrast to previous studies, vascular lesions were included in our approach, and that is often a set of data that others have not incorporated. When deriving cognitive resilience, again, we use linear regression. We get the residuals from there. We did some weighting based on the design of the ERIC study, imputed data that were missing on the clinical side, and then standardized cognitive resilience. And then we used regression to identify proteins that were associated with cognitive resilience. Ultimately, we identified 30 proteins, most of which were associated with positively associated with cognitive resilience, and a subset that were negatively associated with cognitive resilience. Importantly, some of these were only identified by analyses that used sex and race interactions. So some of these proteins were only identified, for example, in women, which is also in line with some of the work in cognitive resilience, where there's some evidence for sex effects when it comes to the impact of cognitive resilience. We identified a number of pathways and molecular networks using ingenuity pathway analysis, and just as a snapshot of what this kind of data has taught us, we see that PI3K is an upstream regulator of cognitive resilience, possibly playing in the insulin resistance. Interestingly, there was a study published in February this year that also showed the same pathway being active for cognitive resilience in humans. And we also see some downstream effects on APP and cytokine signaling, which could be related to amyloid beta clearance. There were a subset of our proteins that were involved in the immune response, and in this analysis, as well as other post-regression analyses, the immune response in the brain ended up popping up very often, and this, as well as some of the single-cell studies that were shown on Sunday, continue to emphasize the role of microglia in cognitive resilience. And the other benefit of doing some of these analyses is bringing up pathways that were proposed before but haven't been the focus of recent work. So one, for example, is the DHCR24 signaling pathway, which is involved in cholesterol biosynthesis. It was described in relation to cognitive resilience in the early 2000s, but recent work hasn't had a focus on it. So again, this work and hopefully other studies can help emphasize that some of these pathways are worth reevaluating. So in conclusion, we identified a set of proteins, networks, pathways that were associated with cognitive resilience. Some of these analyses, some of these associations, though, were sex or race specific, and this emphasizes the value of increasing diversity in your sample, as well as considering sex, especially in the context of studying cognitive resilience. The main themes that came out of our analyses include neuroinflammation, glial cell activity, and synapse. I would refer to the main paper to see more about that. And we also saw some data that emphasizes the importance of looking outside the brain when it comes to cognitive resilience, and potentially some signals that were strong in the kidney and liver. And as far as future directions, one thing we are looking at right now are other platforms. New LISA in particular has been potentially a very important platform to think about when looking at plasma and some of these biomarkers. We're also looking in CSF and looking in other cohorts as well. So in conclusion, this study shows some of the value of looking at a community-based cohort with white and African American individuals and incorporating multimodal data to study cognitive resilience, and I hope that others would join me in using their omic data to study this. So thank you for your attention, and I'd be glad to answer questions at the end of this. Thank you.
Thank you very much, Alvin. We're now going to go to our virtual speaker, who hopefully will be joining us online. This is someone I've known since we were both postdocs some time ago. He's now at Maastricht University in the Netherlands, and his name is Ehsan Pishva. Be talking about epigenomic subtyping of late-onset Alzheimer's disease reveals distinct microglial signatures and molecular inflammatory phenotypes.
Hello, everyone. Thank you for being here. I'm Ehsan Pishva. I lead the Dementia Systems Biology group at Maastricht University in the Netherlands. Today, I will be sharing with you our latest research on epigenomic subtyping of late-onset Alzheimer's disease. I'm sure that you're all in this room aware of the considerable heterogeneity on clinical manifestations of Alzheimer's disease, including age of onset, rate of progression, neuropsychiatric symptoms, and of course, response to the medication. So it is very important to decode this heterogeneity at the molecular level to be able to have precision diagnostics and treatment, of course. In the recent years, there are some omics-based molecular subtypings of Alzheimer's disease. As you can see, two of these studies based on CSF proteomics and transcriptomics across the different brain regions, that they have revealed a very important information about the domination of some biological processes in a subgroup of patients with Alzheimer's disease. So we are using in this study epigenomic data in the same way to identify the subtypes of Alzheimer's disease based on the methylation profiles. Epigenetics or epigenetic mechanisms such as DNA methylation or histone modifications are the mechanisms that without changing the sequence of the DNA, they can influence the transcriptional outcome. Why they are good molecular phenotypes for investigating the heterogeneity in Alzheimer's disease, because it's very well known that they are heavily under influence of the environment and the genotype. As you can see, the genotype can lead to the differential expression in the different brain regions, but also it is known that the environmental factors such as smoking, physical activity, diet, etc., can change the signatures, epigenetic signatures, and then health and disease can be determined by that. So in our previous work, we showed that multiple methylation profile scores as a proxy of lifestyles and modifiable risk factors for dementia. They can play as a predictor of the dementia, where you can see that some factors such as low education and physical inactivity that were measured by the DNA methylation could predict prospective development of dementia, both Alzheimer's disease and Parkinson's disease. So for this study, we used cortical brain samples, prefrontal cortex, dominantly across the 3 independent cohorts of UK Brain Bank Network, Pittsburgh, Alzheimer's Disease Research Center, and RoseMap. In total, 831 case and control. Definite Alzheimer's disease were established based on the presence of dementia, which was measured using CDR and MMSE. And of course, the presence of the hallmark pathology of tau and amyloid. So the methylomic profiling was performed using the EPIC arrays for UKBBN and Pittsburgh, and for RoseMap 450K arrays were available. However, to reduce the dimension of the data, we stick to the most variable data that we use, the median absolute deviation as a measure of variance. As you can see that for the EPIC 320,000 CPGs were selected, and the available one on 450K was around 150 that we used for RoseMap data. So then we moved on to our clustering methods. We used 2 hierarchical clustering and k-means as a non-hierarchical clustering method. The optimal number of clusters were determined across the 2 methods and across all the cohorts using elbow method and normalized mutual information. We identified 3 clusters as the optimal number of the clusters. As I mentioned, that we use both EPIC arrays, which quantifies 850K and 450K in RoseMap data. It was important that we establish that the clusters that we identify using EPIC and 450K are comparable. So we perform WGCNA on the EPIC array data, and we test the correlation between the eigenprobes and the identified clusters. And then the second test, so we subsetted the CPG sets to the ones that they are available only on 450K. As you can see that the value of the eigenprobes when you are using the EPIC probes and the subset of the 450K are equal, and also they can distinguish between the clusters clearly for the sum modules. So now we have generated the clusters across the different cohorts. Now it is important that we evaluate that if any of these clusters are very similar to a cluster in an independent cohort. So basically we have to pair the cohorts and test them that if they are highly similar, and we can call it cross-cohort replication, basically. So we have done this at the two levels of CPG level and sample level. At the CPG level, we check the correlation of the median value of all CPGs per cluster. As you can see that there are two blocks of the three clusters across the different cohorts that they show that basically two subtypes can be established by the cross-cohort replication. At a sample level, what we have done, we performed the SPLSDA, and then we reduced the dimension of the samples, and then using the first two components. Once, for example, we use UKBBN as a discovery, and then we plotted the first two components, and then we projected the other cohorts, means Pittsburgh and Rosemap, on the space of the discovery, which was UKBBN. And then we draw a hole, and then we looked at the overlap of the samples for the clusters for the different cohorts, and using hypergeometric tests, we could evaluate that where is the maximum spatial overlap of the samples. Then using these two methods, we could establish basically two subtypes of Alzheimer's disease, late onset, when we pool all the samples together and we perform PCA, principal component analysis, and then we color labeled the identified subtypes. We could see very nicely the distinct space of these two. It is important because it's completely PCA is independent of our previous steps that we have performed. The next step was that to perform the subtype specific EVOS. So far we were in the literature having the Alzheimer's disease EVOS or pathology Alzheimer's EVOS. So it is important now to perform the subtype specific. We have identified a very distinct signature of the subtypes, which there was some similarity in subtype 2 and the overall, but the subtype 1 is very, very distinct. So we were wondering that what is the contribution of the cell types for this methylation subtype. So the signature of each subtypes from the EVOS was subjected to a tool that we developed in parallel with this study as a cell type enrichment. We used the FANS data, the sorted nucleus data, and then we had the EPIC arrays on the 4 neuron oligodendrocytes, microglia, and astrocytes, and then we generated the panel of the cell type specific, and then we tested the overrepresentation of the signature of each subtypes. And interestingly, both subtypes at the highest level of specificity, they could show the overrepresentation in microglia, and interestingly, the signature of this microglia enrichment were only overlapped one CPG between these two. So it's a quite distinct microglia signature we have identified. The genetic co-localization, so we have found some AD risk genes that they also regulate the methylation signature of each subtypes. You can see that BIN1, we found evidence for the co-localization of the risk variants and the subtype 1 and SPI1 with subtype 2. We had bulk transcriptomic data for a large subset of the samples that we have metylomic data. So we performed the differential expression analysis, again, subtype specific. You can see that 162 genes in association with S1 and 277 with S2. Performing the gene ontology analysis gave us the most interesting results, that you can see that S1 is enriched in immune response pathways and S2 is enriched in the synaptic function pathways. And that was very, very important finding for us. We also looked in the single cell data. We performed the subtype specific single cell microglia transcriptomic data analysis using the Rosemap samples. As you can see that for the 6 microglia, 7 microglia states, again, the overlap of the differentially expressed genes in associated is minimal. So we have a very distinct microglia state association between these two subtypes. And you can see that the immune response activating pathways and innate immune response genes were the most discriminating between these two pathways. So from this, and looking at the individual genes and the direction of genes, we concluded that the load S1 is the more related to innate immune and pro-inflammatory profile of the microglia, and load S2 is more related to anti-inflammatory and resolution. Comparing with the previous one, so we could replicate 2 out of 5 subtypes of both CSF proteomics and transcriptomic data, which was very reassuring. The summary is that we identified 2 distinct subtypes with a very distinct signature. Microglia is the driver of both subtypes, but distinct. We have found some evidence of the genetic co-localization with the methylation signatures, and the transcriptional signatures led us to this pro-inflammatory and the anti-inflammatory signature of them. So given the fact that all the samples were age, pathology, sex, everything matched the AD control, and still we find some sort of stage-related states of pro and anti-inflammatory tell us that, like the AD samples, even at the advanced level, can also have this dynamicity between the anti-inflammatory and pro-inflammatory, which is a known phenomenon, but we could nicely show it here. I would like to thank you all for your attention, and my team at Maastricht University, and the major collaborator, Professor Katie Lennon and her team at University of Exeter. Thank you very much.
Thank you very much, Ehsan. We'll move on now to Giulia Pegoraro, who's from the University of Exeter, who's going to present to us a talk titled Integrated Transcriptome-Wide and miRNA Sequencing Analysis of Systemic Infection in Alzheimer's Disease Brain.
Good morning, everyone. I'm really excited to be presenting part of my PhD project that focuses on systemic infection in Alzheimer's disease brain. So I have nothing to disclose. So I want to start by asking, what if I ask you, what happens if a patient with Alzheimer's disease contracts an infection? I'm sure some of you might say that the rate of cognitive decline increases, or that the rate of mortality of the patient might increase. But what are the molecular mechanisms that are involved in infection during Alzheimer's disease? So this is what we've been trying to answer with this project. But before showing you my results, I want to talk a bit about inflammation in Alzheimer's disease to give you a bit of context. So as many of you might know, neuroinflammation is one of the main hallmarks of Alzheimer's disease, and is characterized by the activation of microglia and astrocytes in the brain. So one of the causing factor and exacerbating factor of neuroinflammation has been reported to be systemic inflammation, which is characterized by the increase of circulating pro-inflammatory cytokines within the brain due to infection or chronic illnesses. So one of the hypotheses is the fact that these pro-inflammatory cytokines are able to pass through the blood-brain barrier, causing the activation or prolonging the activation of microglia and astrocytes, which then leads to a chronic inflammatory state that may lead to an increased neurodegeneration. So I've mentioned infection as one of the causes of systemic inflammation. And there have been different studies that highlighted that not only infection can increase the rate of cognitive decline in patients with Alzheimer's disease, but can also increase the risk of developing dementia in elderly individuals after an infective episode. And particularly, the most common cause of death in patients with dementia has been reported to be bronchopneumonia, a respiratory infection. So what we aim to do in this project is to identify the molecular mechanism that are influenced by systemic respiratory infections in Alzheimer's disease brain. So to do so, we used a cohort of 202 post-mortem prefrontal cortex samples divided in 4 groups, as you can see in the box on the left. So we consider respiratory infection as cases of infection and patients that have been reported with respiratory infection at the time of death. So from the differential expression analysis between these groups, we were able to identify FDR-adjusted results from 2 comparisons. So one between the cases of Alzheimer's disease and the controls, and the other between the cases of Alzheimer's disease with respiratory infection versus controls with a respiratory infection. And particularly, I want to briefly highlight 2 of the genes that have been identified in this analysis, both upregulated in both comparisons, but with different expression levels, which are ADAMTS2 and ACUP6. Which both have been highlighted in other studies to be related to Alzheimer's disease and infection. So for the purpose of this analysis, we focus particularly on the genes identified between Alzheimer's disease with infection versus control with infection. And we look at the enrichment, the cell type enrichment of these genes. And we found results in astrocytes. So astrocytes were the cell type where they were significantly enriched. So we then wanted to understand the broader gene expression network. And for that, we perform gene network analysis using WGCNA. And you can see the results on the left in the heat map. So we identify these modules that are labeled arbitrarily with different colors. And the one that was particularly interesting for us was the yellow module, which correlates both with Alzheimer's disease and infection. So looking at the cell types where these genes were enriched, we saw, again, astrocytes coming up as enriched, but also endothelial cells and pericytes. So we then wondered what was causing these transcriptional changes. So we look at microRNAs extracted from the same cohort and identified FDR-adjusted results in the same comparisons that we did for the mRNA. And one microRNA in particular resulted to be quite interesting for us, which is microRNA 3753P. This was quite interesting because we did not find this gene to be significant in the other comparison, so Alzheimer's disease versus control, but also looking at a meta-analysis of more or less 1,000 samples that, again, look at the differences between Alzheimer's disease and controls, we saw that this gene did not result to be significantly differentially expressed. So then we look at the targets of these genes. We identified targets and then clustered them based on shared gene ontology biological processes and look at the cell type enrichment of the genes within each module. So I want to highlight a couple of modules. If you're wondering why M1 is not present, it's just because it didn't have any significant results. So M2 was quite interesting as it's highlighting biological processes related to viral function. And then the other one that was quite interesting for us was M5, which again is pointing at vascular biological processes and is enriched in endothelial cells and pericytes. So we wondered, did any of the differentially expressed genes that we found in infection overlap with the gene that we found as targets of this microRNA? And we did find them. So most of them have been highlighted to be present in the M5 module, which has vascular biological processes. And then we also look at the genes that we obtained from the gene network and found that most of them were also overlapping with the M5 module. So what we can conclude from this study is that we think that mRNA 3753P may be a key regulator of systemic infection in the context of Alzheimer's disease. And that looking at the overlap between the differentially expressed genes between Alzheimer's disease and controls and targets of the identified microRNA, there is an involvement with endothelial function. And finally, the cell type enrichment analysis that implicated endothelial cells, pericytes, and astrocytes suggested that infection may impact the blood-brain integrity and the neuroimmune interaction in Alzheimer's disease. So future direction for these studies would be to integrate genotype and DNA methylation data extracted from the same samples to have a broader picture of what is happening. Finally, I would like to thank everyone that has been involved in this project, our collaborator, my founders, the UK Brain Bank that gave us the possibility of use these samples. I would like to thank you for your attention, and ASE for giving me the possibility of presenting my work and also funding my attendance here at ASE this year. Thank you.
Thank you, Julia. And just on to our last speaker now, Michael Lutz from Duke University, presenting on molecular insights into shared and distinct etiologies of Alzheimer's disease and major depressive disorder at single nucleus resolution.
Good morning, and thank you for the introduction and the opportunity to present this work. Photography is welcome. Video and audio recording, however, are prohibited. I have no disclosures. Funding is in part from NIH and NIA. The relationship between depression and Alzheimer's disease is complex, and studies have addressed epidemiology, genetics, and underlying biochemistry and physiology that may be shared between neuropsychiatric and neurodegenerative diseases. Depression can occur at 3 different stages in relation to the process of neurodegeneration in AD. Depression can be a predisposing risk factor occurring before the onset of AD pathology. It might also be an early sign of neurodegenerative changes or a prodromal symptom with or without cognitive deficits. Finally, it may occur at the more advanced dementia stage of AD. We and others have studied the shared genetic etiology of Alzheimer's disease and major depressive disorder, or MDD. The Mendelian randomization analysis in this study showed a moderate influence of SNPs associated with late onset Alzheimer's disease on the risk of MDD with an odds ratio of 1.4. Phenotype definition needs to be carefully specified and can range from longitudinal analysis of depressive symptoms to diagnosis of MDD. Consideration of confounders, including age and sex, are critical for these studies. This study examined shared mechanisms across major psychiatric and neurodegenerative diseases. The study integrated GWAS results with human brain transcriptomic and proteomic data. The specific result on this slide is genetic correlation between neurodegenerative diseases, which are the rows, and psychiatric diseases, the columns, based on linkage to equilibrium score regression. There's a highly significant correlation between AD and MDD for one study, but a weak negative correlation for the other. Differences between the studies are in part that AD1 is a very large study of greater than 400,000 individuals that included data from the UK Biobank as proxies for AD or controls. The AD2 study is based on a clinical diagnosis for AD, again a large study but no proxies. In previous work, we identified shared risk loci between AD and MDD. Another study using bulk brain transcriptomic data identified sex-dependent differentially expressed genes, or DEGs, in AD comorbid with depression, in contrast to AD without depression. In the current study, we investigate cell type-specific differences in gene expression from autopsied brain tissue from Alzheimer's disease patients with and without MDD, and contrast them with similarly aged individuals and also with young individuals. We identified common and different transcriptomic signatures, differentially expressed genes, biological pathways, and cell communication networks. The design of the study and analytical approach are shown here. We constructed 3 cohorts to create contrast between individuals with and without MDD, young individuals, and older individuals with AD. For pre-processing, we utilized Seurat's SCT transform for normalization, integration, and clustering. Importantly, to put all of the cell type annotations on the same page, we annotated the data sets with the SC Mayo map. Differential gene expression was performed using the Nebula program, followed by a 2-stage gene selection process to determine gene overlap between the relevant groups. Pathway analysis was completed using Metascape, and cell communication networks were derived using CellChat. This slide shows the cohort demographics. The 2 older cohorts were derived from the Rush University Medical Center's ROSMAP cohort. Average ages are in the mid to upper 80s, with approximately 70% female. The AD patients were slightly older than the cognitively normal controls. The young cohort had an average age of 43 years and was approximately 50% female. The references provide more detailed information on the cohorts and samples. This slide shows the number of differentially expressed genes, or DEGs, by cell subtype for the AD cohort. The differential calculation is for presence of a diagnosis of MDD. The highest number of DEGs, slightly over 200, is for the GABAergic neurons. There are around 50 DEGs identified for glutamatergic neurons and microglia. Fairly similar overall numbers for the older cognitively normal cohort. As with the AD samples, the highest number of DEGs, slightly over 200, is for the GABAergic neurons, and interestingly, an almost equal number of DEGs for the glutamatergic neurons. Large numbers of DEGs were identified for the diagnosis of MDD for the young cohort. As with the other cohorts, some of the largest numbers of DEGs were observed for the GABAergic and glutamatergic neurons, with relatively fewer DEGs identified in microglia. This slide shows common and distinct glutamatergic DEGs, contrasting the older cognitively normal group with the AD group. The Venn diagram shows the numbers of unique and common DEGs, in this case, 14 common. The table is for the common DEGs. Fold change in FDR p-values are the comparison of the presence and absence of MDD. Fold changes generally have opposite signs for the 2 cohorts. The highlighted rows show overlap with a second comparison between samples from the young and the AD groups. We'll discuss that one on the next slide. The rows highlighted in pink co-occur in both comparisons, while the rows in blue are unique to this comparison. The specific rows chosen for highlighting are based on potential biological significance to MDD and AD. ARC is a master regulator of synaptic plasticity. CCN1 promotes cell proliferation, chemotaxis, angiogenesis, and cell adhesion. DUSP1 dephosphorylates MAP kinase, MAPK1, ERK2. This protein plays an important role in the human cellular response to environmental stress, as well as in the negative regulation of cell proliferation. JUNB is activated in response to various stimuli like stress, inflammation, and neuronal injury. Here we see the common and distinct glutamatergic DEGs contrasting samples from the young and the AD groups. There's 7 common DEGs. Fold changes generally have the same sign or direction and actually the same relative magnitude for these completely independent cohorts. Rows highlighted in pink co-occur with the older AD comparison that I showed on the previous slide, while the rows in blue are unique to this comparison. SERPINE2 is a serine protease inhibitor with activity towards thrombin, trypsin, and urokinase that promotes neurite extension by inhibiting thrombin. JUN is activated in response to stress. With more time, I could discuss the GABAergic results. There were similar numbers of common and unique genes, and several genes for GABAergic pathways were identified as FDR significant and differentially expressed, including the GABA-RA6 gene. This slide shows the Metascape pathway analysis for the DEG results for the glutamatergic neurons based on the AD cohort, comparing samples from individuals with and without MDD. The AP1, or activator 1 pathway, is downregulated in individuals is downregulated. Components of this pathway are transcription factors that regulate gene expression in response to stimuli, including stress and cytokines. There's an intersection of these pathways with the MAPK pathway. AP1 is involved in the regulation of several biological and cellular processes relevant to AD and MDD, including apoptosis and inflammation. Only 2 upregulated pathways were identified in the analysis, very broad pathways for epigenetic regulation of gene expression and chromatin remodeling. Microglia are of interest for their role in the development of neuropsychiatric and neurodegenerative diseases. The largest number of microglial DEGs were from the contrast of the young cohort with the older cohort, with 13 common DEGs. Genes include transcription factors, transporters, and scaffold adapter proteins, including PRAM1 and SNX9. PRAM1 functions include myeloid differentiation and lipid binding. SNX9 is involved in endocytosis and intracellular vesicle trafficking. Pathway analysis in the microglia focused on the downregulated genes in the AD cohort. So in this case, downregulated is MDD relative to no MDD. Several pathways are implicated in both AD and MDD pathophysiology, including the PPR signaling pathway, interferon alpha-beta signaling with an impact on neuroinflammation, and VEGF-A, VEGF2 signaling. These cell communication networks are for the number of interactions between the cell subtypes. The chord diagram on the left is for AD samples. The diagram on the right is for the older cognitively normal samples. Blue arcs indicate increased numbers of interactions, while red arcs are less in the samples from individuals with MDD relative to no MDD. Overall, for both plots, we see a decrease in CCC for the MDD group compared to controls, since there are numerous red arcs. For samples from individuals with MDD, comorbid with load, in contrast to load patients without MDD, there are notably fewer and weaker interactions between the GABAergic, glutamatergic, and interneurons, as shown by the thicker red lines. The blue arcs indicate an increase in CCC for several pairs in the AD sample group, notably GABAergic neurons and microglia, and glutamatergic neurons and Bergman glial cells. This pattern is not observed in the cognitively normal sample group, where there's an overall net decrease in CCC for these pairs. In contrast to the number of interactions, this chart shows the strength of interactions between the cell subtypes, with blue increased strength and red decreased strength. Again, AD samples on the left, cognitively normal on the right. Notable aspects of this plot are the increase in the strength of interactions between GABAergic and glutamatergic neurons for the AD samples relative to the cognitively normal samples. Again, this is for MDD cases relative to no MDD in both. In conclusion, the single-cell analysis of brain tissue provides mechanistic insights into the shared and divergent molecular etiologies, dysregulated pathways, and impaired cellular communication networks between load and MDD. Future plans include investigating other comorbid NPS, including apathy and anxiety, replication. The PsycAD Atlas will be a great resource for this, and delving into some of the biology that was highlighted from the analysis. I want to acknowledge the collaborators on this project, Dr. Ronit Chibafalik, Joey Mann, and Dan Gingrich. I also want to express my appreciation to David Bennett at Rush and Gustavo Turecki and Karina Nagy at McGill for their help with the data sets, and also funding from NIH. And finally, I wanted to highlight a research topic organized by Frontiers that's of potential interest to the multi-omic community on precision medicine and neurodegenerative diseases. This topic covers mechanistic understanding of AD through translational studies to develop biomarkers for early AD diagnosis. There's an opportunity to submit papers and participate in an ongoing series of webinars. The QR codes provide links with more information. Thank you for your attention.
Thank you, Mike, and thanks to all the presenters for the very intriguing, very interesting talks. We're now going to open the stage, the room for questions. We'll start with the in-person here. So number 6, please.
Hi, good morning. My question is for Julia. Great work. As someone who, when first started, would associate GFAP, for example, with inflammation and then realized I was totally wrong and shouldn't immediately jump into seeing an astrocyte being active as meaning inflammation. And so many great people working in astrocytes, I think, would totally argue about that. And then you come and you show us that when there is inflammation, you do see astrocytes doing something. So my question for you is, what does it mean? Why are the astrocytes responding, and what are they doing in that response?
Yes, hi Isabel. Thank you for your question. So our current hypothesis is that we think we're seeing astrocytes in coordination with endothelial cells and pericytes. So it might have to do with the blood-brain barrier. So we know that astrocytes are, the handfeeds are part of the blood-brain barrier. So what we are thinking is that there might be some deregulation or the integrity of the blood-brain barrier is compromised. And for that we see astrocytes. And then there is one other part of the results that is not here. So when I was looking precisely at the astrocyte subtype, I was seeing in the differentially expressed genes in infection, I saw that the subtype was reactive astrocytes that is usually deregulated in neurodegeneration. So yeah, is a great question, and we are trying to figure out the same, but this is our current hypothesis on that, so the blood-brain barrier might have something to do.
Thank you.
Thank you. Number 4, please.
Nilüfer Container, Mayo Clinic. I also have a question for the speaker who presented on the upper respiratory infections and brain transcriptional changes. Given that you saw some changes that could implicate a problem with the blood-brain barrier, have you looked at the presence of microorganisms in the brain transcriptome? And can you share more information about the respiratory infections? What microorganisms were involved, and whether you measured for cytokines either in blood or CSF?
Yes. So thank you for your question. This is a great question. So we also wonder if we were able to identify microorganisms within the brain. So we did try to look for that, but unfortunately that analysis didn't yield any significant results. As for the type of infection, unfortunately we are quite limited on the information that we have from the clinical reports. So not always we know the microorganism that is involved in the respiratory infection.
Thanks.
We've just got a question online, and then we'll come back in the room because we do have time. So this is for Pablo, and it's from Courtney Sutphen from Wake Forest University. So for the variation in PC2, is it possible that instead of blood-brain barrier breakdown, that those signals may be related to CSF sample quality, for example, blood contamination?
Yeah, we tested this hypothesis as well. Thank you for the question. And we did observe a mild association with the red blood cell count in the proteomics PC2. However, once we adjusted by the CSF turbidimetry, the CSF total protein content, this association was lost, and also in comparison, the huge signal that we got for CSF turbidimetry, the red blood cell count signal was really small. So there might be a small contribution, but we think that most likely the phenotype is caused mostly by BBB disruption.
Thank you. And back to the room, number 5, please.
This is Adeline Tsai from Mayo Clinic. Thank you all for the talks. I have a question regarding the ARIC study. So I'm wondering, do you know like the percentage of, among the participants, the percentage of people who have ApoE4, like who are ApoE4 carriers? And if you have also considered or have done analysis to look at like the interaction between sex, race, age, and ApoE4 status?
Thank you for the question. So I believe it's around 30%, if I remember correctly. But I have the data if you are able to come up later. So one of the important things about cognitive resilience that we've seen in this study, as well as in RoseMap and a few other cohorts, ApoE4 carrier status and cognitive resilience are perpendicular, meaning that they're not associated. And that's partly by, it's both empirically and partly by design. The concept of cognitive resilience is distinct, essentially, from the risk, the genetic risk for Alzheimer's disease. So in these analyses, we didn't do additional stratification by ApoE because it wouldn't have made a difference, as they're not associated with each other. But the point about race, sex, and ApoE4 is well taken, thinking on a larger scale, especially with greater diversity in our sample. This sample size is still relatively small, I guess, to really get at that question.
Okay, thank you.
Number one, please.
This is Pan Zheng from Uncle C4. This is a question for Dr. Michael Lutz. So usually we group all this neurodegenerative disease, AD, Parkinson, Huntington, because they have a misfolded protein. All this protein aggregate. But then in depression, there's the same kind of protein aggregate?
Yeah, thanks for the question. It's a good question. And yes, there are clearly misfolded proteins. Really, the design of this particular study was to look at specific cell type resolution and not the misfolding of the protein. So your question is kind of getting to some of the mechanistic pieces. And as far as depression, even going so far as MDD, really it would not be a study that would answer kind of a question on whether or not there would be misfolded proteins involved.
So in the AD GWAS, in chromosome 19, they actually really have hundreds of a hit over there.
For
the
ApoE.
Right. But then how about the MDD? They also have a certain GWAS in the chromosome 19.
Yeah, some of the studies I cited earlier, there are some large GWAS on MDD from some of the consortium. I don't believe that there's a particularly strong signal for ApoE, though. I know, Oni, do you?
Not exactly ApoE, but just the chromosome 19, like a Q13, that location.
I don't believe there's a particularly strong signal.
Okay, thank you.
Okay. Number 6.
Yeah, I have another question for the ARIC study. Am I correct that you looked at current cognitive function?
Yes.
So here's what I'm thinking. There's a lot of variability in cognitive functioning long before any Alzheimer-related pathology. So since there's a lot of longitudinal data in the ARIC study, would it make more sense to think about cognitive resilience in terms of change in cognitive function from prior levels rather than just current level of cognitive function?
Thank you for the question. So the ERIC study was started in 1989, and the first cognitive measure was at visit, we had one at visit 2 and visit 3, so like in the 90s, and then there was a large gap of time before visit 5 came in 2011. So I did do some longitudinal analyses using those 3 sets of cognitive data. But essentially, the longitudinal models did not perform, there wasn't as much variation given the kind of distance between midlife versus late life cognition and our single measure of MRI, because we only had MRI starting at visit 5. Your question, though, is extremely important. My colleague Maud at Rush has developed a number of methods to look at longitudinal cognitive resilience, and she's published a few papers on that. I know a few other studies have also performed measures of cognitive resilience longitudinally. I believe those are better methods, and that's what I hope to use now that we have some post-visit 5 data. So ERIC is, I think, doing its like 10th or 11th visit right now. So since visit 5, we've done a lot more MRI and a lot more cognitive testing. And so the hope is we'll have the data now in order to do exactly what you propose and look at it longitudinally.
Well, just to follow up, but I think the key is the cognitive function before the pathology. So if you're not seeing the effect when you're looking at change from those earlier assessments, it may raise questions about what you're finding in terms of resilience or lack of evidence for it.
Right, and when I looked in the midlife to late life range, essentially there wasn't a lot of interperson variation. Within individuals, our cognition will fluctuate, but not to the degrees we would see large differences in the psychometric battery across the whole population. So I completely agree that is that's a big part of what cognitive resilience is. This study didn't have the best data to get at that question, but other cohorts I think do.
Okay, thank you. Number 4.
This is Marco from Brazil. My question is for Julia. I was wondering, how did you do the cell enrichment for your analysis?
Yes, so thank you for your question. So for the cell enrichment, we use a method which is called EWC. So it's a computational method where you have a reference data set, in our case, single nuclei of the major cell type in the brain, looking at prefrontal cortex, and then you perform an enrichment analysis on it.
Thank you.
Okay, we'll take another question from 4.
Hi, good morning. Natalia Vilor Tejedor from Barcelona Beta Brain Research Center. Brilliant session. I'm impressed about all the amazing work that you are doing. And actually, I have a general question for all of you related to the dynamics of the multiomics data that you are using. So in most of your studies, you are showing cross-section or one-time point analysis. And I want to know, what are the challenges and what do you think the longitudinal dynamics of this multiomic data can improve or provide more additional information in your studies?
Since I was just talking about longitudinal data, one area of work that I've become increasingly interested in when I'm looking at the kind of the natural history of cognition and of neuropathology is how these biomarkers change from, especially during MCI. So some of the imaging studies have shown that there's like an inverse relationship between the biomarker and cognition. During the MCI phase, the theory being that the brain is kind of fighting back, right, a little bit more, and then at some point the dam is broken, and then you see kind of the relationship you might expect a priori. And this is, going back earlier to this idea of longitudinal data, this is something that I want to think about a little bit more, especially for the plasma data. When you're looking at neurodegeneration, once the neuron is gone, it's gone. But for the plasma and for the CSF biomarkers, it can go in either direction, and the disease phase also might be really important. And I think this has been, especially in the study of cognitive resilience, an underappreciated factor, and really emphasizes why the cross-sectional design might help us start generating some of these hypotheses, but really we need more longitudinal data. And unfortunately, that means longitudinal for each of those things. For me, cognition, imaging, and the biomarker, the omic layer as well.
Yeah, I would just add, again, in terms of definition of phenotype, the longitudinal data can be absolutely critical and helpful to do a much more exquisite definition of the phenotype. Unfortunately, with postmortem tissue, you get one sample, and that's a snapshot.
Ishan, you want to comment too?
Ehsan, can you hear us online? Just wanted to invite.
Yes, hi. Yeah, I'm happy to talk specifically. So as it was mentioned for the postmortem brain, so it is not a possibility. But for the brain one, since we have been expanded the subtyping of the methylomic data on the blood data, it is quite useful if you have the longitudinal methylation data, given the fact that the DNA methylation can be a proxy of the DNA, the environmental and lifestyle factors. So that would be quite useful to look into the longitudinal, for example, methylation quantification to specifically look at those individuals that sometimes called as reverters, because we have the MCI patients that after a while they turn back to control, and those are specifically can be interesting to look that if there is any improvement in the environmental factors that help them to revert from those clinical symptoms.
Okay, thank you. Thank you. Number 2, please.
Thank you very much for very exciting talks. My name is Sharna Janalidze. I'm from Lund University, Sweden. I have a question regarding CSF variability. Have you tested whether associations between A-beta-42 and p-tau181 with measures of Alzheimer's disease pathology, for example, with amyloid PET and tau PET, are affected to the same extent by a reference, top reference protein in comparison to A-beta-40, for example?
Yeah. So thank you for the question. Regarding the first question, unfortunately, we didn't have PET data for these samples. It would have been great to do these comparisons. And actually, there was a study I mentioned in the first slide, published during last year, and they did a quite similar approach to us, and they did find that they were able to increase the PET and CSF concordance, especially in those subjects with kind of around the threshold levels in the CSF. And regarding A-beta-40, it's also an hypothesis we wanted to explore. The only problem was that we only had available measurements for about half of the samples, so we couldn't perform a full assessment. But the beta-40 is actually a quite good biomarker for CSF clearance. And there's also a lot of studies coming out now proposing to adjust also p-tau levels, for example, for A-beta-40. So A-beta-40 is a good biomarker for CSF clearance, of course, but our results indicate that pretty much you can use a whole amount of proteins because there is a lot of proteins that are strongly affected by this phenotype. But yeah, A-beta-40 does this as well.
Thank you.
Okay. Any more questions from the room? If not, we will close this session. Thank you all for attending, and thanks again for the speaker for the wonderful talks. Thank you. And enjoy the rest of the conference.